Today we have with us Dr. Ukabali Baba Wazir from Kashmir. Sir is board member of Department of Microbiology, University of Kashmir. One very good thing I would like to point out, Dr. Ali has received Young Scientist Award in 2021 by Microbial Society India, and he has received a Best Poster Presentation Award in 13th Jammu and Kashmir Science Congress in 2018. Sir is currently working as lecturer in Model Government College, Charare Sharif, Kashmir. Dr. Ukab has cleared UGC NET examination in 2019, ASRB NET, which is Agricultural NET, in 2016 he has cleared, and he has also cleared GATE 2020. Dr. Ali has attended many international conferences in the subject of biotechnology and microbiology. Dr. Ukab has published more than 15 national and international research papers in various reputed peer-reviewed journals, and also he has published more than 8 book chapters in various national and international publishers. Sir has done his PhD from University of Kashmir in 2020. He has completed his MSc in Environmental Science from Central University of Jammu in 2014. And one very nice thing I would like to point out is Dr. Ali has also completed his B.Ed from Kashmir University in 2016. Now, I welcome our today's lecturer, Dr. Ali. Sir, please, you may start your lecture.
Thank you, Jai sir, for such a nice introduction. And I thank Microbiology Society India for providing me an opportunity to present my PhD work again. And today I feel like I'm again a scholar and doing the job to justify my PhD things. It's always nice to talk about the PhD work which we done. And Microbiology Society is very eager for this purpose that researchers throughout the India came in a single platform to discuss their work, to see what can be next done in their work and what can be the lacunae which has been in the PhD, which can be taken for the postdoc. Thank you, Microbiology Society India, for providing us the opportunity. And I thank Dr. Arvind Deshmukh sir, Sarvanjit sir, that the way society is going, I hope that this will be a nice platform in coming months. I not say years, in coming months, it will be a nice platform for, especially for the microbiologist students, to see what type of research is going on, what we need to, where we need to, what trend we need to follow, where we have to shift. And this is really amazing. The society is working in a better way for the microbiologists. So, good morning, everyone. I'm Dr. Ukabali, and I have done my PhD from University of Kashmir under the humble guidance of the two renowned scientists. One is Professor Bashir Ahmad Ganai, who is currently the director, North Campus, University of Kashmir, and my supervisor was Dr. Ruqayya Nazir from Center of Research for Development, University of Kashmir. And the topic which I will talk today is about the in vitro sequestration potential of heavy metal tolerant microbial strains isolated from saffron soils of Kashmir. And when we know about the heavy metals, you know that there are number of definitions regarding the heavy metals. We call them highly dense metallic elements among the important class of contamination in the environment. Reason being, these are the widespread pollutants of great concern, because we know they are non-degradable, and thus when we say they are non-degradable, we say that they are persistent pollutants, and that's the problem they will create in the environment. And we know that introduction of metals in various forms cause modification in microbial communities. Obviously, whenever we have some citrus like metal citrus in the microbial in the environment, which have various number of microorganisms, they modify microbial communities. And what we need to do is how we can decrease. See, there are a number of sources of heavy metals. You see, we have metallurgical processes where we get arsenic, chromium, nickel. We do have plastic manufacturing units. We have electroplating companies. And in the last, we have fertilizers, pesticides, and cement industries. And my topic is based upon a very important plant that is actually saffron Crocus ivus, we call that in botanical name. Reason being that the quality as well as the quantity of this plant, of this spice, is being degraded from various years. We don't have a good bumper crop of saffron. Reason being, a number of cement industries has been established in the area of the Pampur, which is the Kareva area, which is actually the land needed for cultivation of saffron. And the number of industries, cement industries, is being increasing day by day, and the quality as well as quantity of this important spice is being degraded. And you know that heavy pollution, heavy metal pollution, growing concern nowadays. We don't have to say about this. And India is no exceptional to this pollution. And Jammu Kashmir in particular is facing the same problem. They have the pollution through a non-prominent soil types, and so far as report is gaining importance now. Once again. And the source here mainly remains in the form of cement manufacturing units. As I mentioned, this is a publisher. There's a publishing data about this, the heavy metals impacts, unscientific, intensive agriculture practices, various solid waste dumping sites, e-wastes thrown in an open field, and even the application of wastewater sludge into agriculture fields are some of the important sources of heavy metals which are nowadays available to us. And research is going on all over the microbes. Microbiology is nowadays a trending subject throughout the world, throughout the globe. But a meager work has been done in the soils of Kashmir Valley, and that's why the impact of heavy metal on saffron, that's Crocus sativus, which is an important cash crop of Kashmir. You know that this part of Kashmir is only known for the tourism as well as for the agricultural activities, and saffron being the backbone of our economy. And if the quality as well as quantity will go down, this will badly impact the economical conditions of the state. And keeping in view the economical importance of economical cash crop in the region in mind, a comparative and elaborative study was taken for observing the metal tolerant competent microbes residing in the saffron soils. And this is an image. This is an image. You see a number of cement industries are being present in the saffron. In the front, you are seeing that these are the saffron lands. In the backyard, you are seeing the chimney where a number of emissions is being taking place from the saffron industries. And study area, we have taken two study areas. One is the area which is being under the influence of the cement industry, and other area is in control site, which is not in the influence of the cement industries. One is from Chirari Sharif control site. There the saffron is also grown, but not in the influence of cement industries. And in Pampur, there is the influence of saffron, the cement industries, and we have used this. You see, these are some sites. You see, site one, it is in the backyard of the site. You are seeing a big cement industry that is lying. Site 2 is also from the cement industry site. Site 3 and site 4 is an area where there isn't any influence of cement industries, and there's a least intrusion of these factories as well. So objectives which we designed it for our PhD was analysis of major heavy metals in the soil, then isolation and character of soil bacteria from soil samples, appraisal of heavy metal, we called that as minimum inhibitory concentrations against various metal ions, and then mass cultivation for metal tolerant strains for assessing their potential at laboratory conditions. The work was simple. First, we go for the site selection and sampling, then heavy metal analysis, isolation of bacteria, MIC, and then the metal uptake of potential at laboratory conditions. And you see the methodology, we have a very simple methodology, that is scientific methodology that is established in the literature. Heavy metal analysis was done with an AES, atomic absorption spectrophotometer. Isolation and characterization was done as per the standard procedures laid by Burgess Manuel et al. I'm not giving much focus on methodology because this is a universal methodology that is being utilized, and it is written everywhere in the literature. We can get it very well. Methods, isolation, we go for serial dilution, split plate and streak plate, then for pure cultures. Identification, that includes some micromorphology like shape, margin, elevation, size, texture. Microphysiology, we did it under the microscope. Gram staining was done, and we used the two techniques for the identification of these bacteria. One is multi-top MS, and second is molecular characterization. In the multi-top, it was done in NCMR Pune, and with the help of a great scientist and Dr. Praveen Rahi in the Praveen Rahi's lab, NCMR Pune. And then the molecular identification was done using the Sigma kit, DNA genome kit from the Sigma, and it was as per the manufacturer protocol, it was done. And as per the sequencing was concerned, the PCR projects were sent to Agri-Genome Kerala and NCMR, and by the sequencing was done and further evaluated and compared to nucleotide and all the sequences which we have got are available at NCBI, and I also submitted these isolates in NCMR Pune. Molecular characterization, we know it's the protocol is very much simple: culturing bacteria, extracting the genome, DNA extraction, PCR application, sequencing, and then identifying using NCBI blast sequencing. Heavy metal was done as per the standard this protocol, and then mass cultivation, and it was done again using the standard protocols for the mass cultivation for assessing potential at laboratory levels. So what we did and what were our results? You see the analysis of major metals, and 4 sites, as I told earlier, were selected, and in the 4 sites we have seen 1 was control and 3 were where there was the influence of the cement industries. Atomic absorption spectrophotometer was used, and that is Perkin Elmer made. That is available in the University of Kashmir. We did this analysis there, and you see, if we go towards the estimation site at Saffron, that you have seen site 1, site 2. These are actually the influence of industries, especially cement industry sites. You see, the as per the Mercury is concerned, the levels from December 16 to March 18 is increasing day by day. And on the right side of this table, I have given the WHO standards. And as per the results of concern, mercury levels have gone up beyond the permissible levels, what WHO is saying, especially in the saffron soils of Kashmir, which is a cause of concern again. And copper is also increasing. Copper levels have gone also up in these soils. This is also an alarming for we people, especially for the saffron growers. Again, if you see site 3 and site 4, as compared to the site 1, 2, and 3, our levels at control site are way down. What is the permissible level that has been set by WHO as per 2005 standards? And you see there is 0.59 milligrams per year, and you see ppm per parts per million. And again, at 3 sites, mercury has gone overshoot the WHO sites. As per the copper, again, this has been overshooted from this what you called as our sites, that's for industrial sites, cement industry affected sites. And just what we analyzed is that our study find that metal accumulation is higher at pamper sites as compared to the control. That's our Sarship site. It was found that the site accumulation heavily mass copper was much, and after that it was manganese, nickel, lead, zinc, chromium, mercury, cadmium, and cobalt. And the same trend while at site 2 and site 4, this was the order of the metals which were present in the soils. In the study, metal accumulation levels are not uniform, and that's obvious. The metal accumulation cannot be uniform, which may be due to factors such as fluctuating environmental conditions like temperature, soil pH, and soil moisture, availability of the elements in the soil. And the concentration of heavy metals were found to be maximum near the cement factory sites, reason being the factory clean and preheating preclination operation of cement factories. Also, the raw material used in the cement production, including secondary fuels, contains the heavy metal. And metal concentration showed declining trend from factory side to the control side. As I said that once we move from the factory sites where the saffron is grown, and once we move away from that towards our control site, then the concentration obviously decreases from this. And we have the increasing level of heavy metals, maybe due to the change in pH of the soil. As a result of each unit decrease in pH, the concentration of metals are approximately doubled. Once the pH of the soil change, it has an impact on the on the metal increase metal concentrations in the soil. The increase in microbial can be due to the cement factory kiln and preheating operation of the cement industries. And you know that mercury, when we say that what are the major sources of mercury, you know that we have some of the pre-kiln activities, preheating and calcination operations, where we use some of the stones, and we also use coal for in these cement industries, and that can be a major source of mercury in these soils. And bacterial isolation, you know, we have isolated bacteria from the 4 sites, 3 from our study site, 1 as well as control site, on seasonal basis from December 2016 to October 2018. From study sites, we have chosen assessed quality forming units on nutrient agar and LB agar also. The soil sampling, we use the CDLW. I will come. A total of 37 bacterial isolates were collected from the control site, and 35 bacterial isolates were obtained from the Pampur site, which is being affected by the what you call as heavy these cement industries. This is the colony-wise quality CFU forming units of bacteria. And from 2016 winter, spring, summer, and autumn, we have done it season-wise and site-wise also. And macromorphological characteristics of each isolate that were identified from these 2 sites, especially 2. One is control and one is our in the cement industry affected sites. And then we have gone, these are some of the glimpses of the pure isolates that have been cultured from different sites. And then we have again the isolates were then subject to gram staining, obviously, because nowadays we have the genomic kits which are based on the gram this nature of the bacteria, gram positive and gram negative. Then you have to follow the protocol as per the grams nature of the isolates. And then we that's why we have to go for the gram staining. And you know that 35% from these gram stains out of 37 from the control side, 35% by gram negative and 64% by gram positive. And in case of the Pampur side, we have 62% gram positive, and we have 37% gram negative. And then we have the Maldita. This is one of the important instruments nowadays available and readily available for an researcher. If even it can identify your bacteria within 3 to 4 hours, and it is a very good instrument. I have done this from NCMR Pune with the help of, I already told, Dr. Praveen Rahi, who in whose name we give. And there is an top score, multi-top identification score, where we have the identification after when we have greater than 2, that's an authentic. We call that as authentic. When we have less than between and below 1.7 identification score, we go that there is some sort of indication, no identification, because there can be what you call as impurities in your culture. The culture may not be pure, and you have to reculture your bacteria again and then do the multi-top identification. And similarly, various species, by all the species were identified using the multi-top MS. And you see, this is the actual the fingerprints obtained of the whole colonies of different bacterial species. This is a multi-top. This is actually a protein profile of these species, which we analyzed it in the multi-top, and based upon this protein profile. In multi-top, we actually extract the proteins of these bacterial pure isolates. Then we put it in the instrument, and then the normal matrix is being used in that. And after that, it is put in the instrument, and we get the protein peaks. And that mass spectra has a library. Based on those libraries, these bacteria are being identified. These are some of the spectra we get from the multi-top. This is also our multi-top gel view pictures of these isolates. And these are representative 16S RNA amplified picture of our DNAs. And this is the species which we have identified using molecular techniques. Then we have submitted it in the NCBI, and we have got the accession numbers for all these isolates. These are available online. I know you know this is the minimum elution phylogenetic tree that is being used using the what you called as mega 10, and that has been used to construct these phylogenetical trees. And 37 bacterial isolates were isolated from Sarishiri, that's for control, and 26 different species among them. And when we count 37 were total number of isolates. Among them, 26 were different species, and these are some of the species which were totally different. And from the Pampur side, which is actually a cement affected side, where saffron is actually grown under the influence of cement, dusts, and etc., we have got 35 bacterial isolates, and among them, 23 species were totally different. And the taxonomical classification of what we got from the control side says that majority of the bacteria were from the Bacillacea and followed by Enterobacteriaceae, then from Planococcacea, Staphylococcus, Iridomonaceae, and Pseudomonaceae. And then we have Pampur, we have again the majority of the bacteria from Bacillacea, then for Enterobacteriaceae, then Planococcacea, and rest. This is the taxonomical classification of bacteria that were isolated from the two sites. And we know that due to extreme importance of microorganism energy and material transfer, microbial diversity in contaminants is extremely important. Nowadays, we don't go for the traditional use, because nowadays we want to be eco-friendly and we have to devise some eco-friendly techniques. And this use of microorganisms in detoxification or sequestration of heavy metals, this is an eco-friendly technique that can be utilized. And you know that productivity, morphological, why we go for morphological identification. Certain features of the isolates are largely depending on identification of bacteria. But at the same time, these phenotypic profiling in gram staining results, colony morphologies, and we have to go for. Sometimes we don't know what if we can get a new species from these sites. And at that time, morphological identification, molecular identification, these go side by side. This is an important, because whenever we submit a new species, then they ask for number of things. And that's why we have to go for morphological. And it's good when we compare morphological identification as well as molecular identification plus new reliable methods, up-to-date methods like Multitask. These give us a reliable data which we can rely upon, and we can communicate our results with high surety that yes, if we have this time of bacteria, new bacteria is being isolated, we are reporting it for the first time, then our results are very much elaborative itself, using large number of techniques and giving the results with proof. It always helps very well. And bacteria belonging to diverse groups in the saffron with Bacillacea, I told you dominant phylum, gram positive more dominant than gram negative, and identification of more isolate, which is concurrence with our study. Some have done that. And on the MIC, see, determination of MH agar, Muller-Hinton agar, and broth mediums during the study period revealed that 49 from all site species, 8 species were tolerant to mercury. From control as well as for total 49 species, 49 isolates were put to MIC, and from that we have got 8 species that are tolerant to this mercury. That is Staphylococcus equum, Lucretia, Verneri, Klebsiella pneumonia, Serratia, Bacillus sapinensis, Bacillus zahanensis, and Pseudomonas putida. And while in case of cadmium, we have isolated 7 strains that were tolerant to cadmium. And again, likewise, we have also identified 4 isolates which were tolerant to the lead. And let me tell you, the range varied from 500 to 700 ppm in our study, and that's at laboratory conditions. And from nickel, we have isolated 5 strains, and the range of that strains varied from 600 ppm to 1000 ppm. This was the 96 well plate, which we used for determination of MIC, using control as well as our species, and then feed them with different concentration of metals. And you see, these are the photographs of mercury tolerant. This is on the agar, and we have also done it on the broth. This is the broth method, which we usually done for the MICs, and then we have also cross-checked our results on the agar, giving the same concentrations. You see, there are a number of strains for mercury. If you look, there are 5 plates in the 5 plate. Now, in plate, this is on the extreme right, there is control, and on the extreme left are the 4 isolates. These are the various concentration of metals. Same is for the cadmium, same is for the lead, and same is for the nickel. And you see, there's a clear zone of clearance. There isn't any growth in the mercury tolerant bacterial strain, you see, as compared to control. And there are some concentrations where the growth is, and on 60 ppm there isn't any growth. And same, we have repeated our results on the what you call as broth, medium MH broth. You see the optical densities which we take for MICs forever after 24 hours of reading, and this is what we did for optical densities. This is for mercury. The highest level what we get was for stability was 300 ppm, and the maximum uh, MIC was for Pseudomonas putida with the highest potential of 400 ppm. It was able to survive on these on the laboratory condition. And similarly, this is a graph of the same. We have plotted for mercury tolerant bacteria. And then we have the optical densities that was based on broth method. And we have seen that it is Klebsiella pneumonia showing us the tolerance of 900 ppm of these metal tolerance on laboratory conditions on the broth, as well as on the what you call as agar medium, MH agar. And same wise, we have seen this is the graphical representation of the same. We have then taken optical densities for lead, and we have seen 900 ppm was the maximum tolerance from Bacillus thuringiensis. And this was very nice to see that it can thrive up to 900 ppm parts per million in the laboratory conditions. And this is again for the nickel, we have seen that 1000 ppm Klebsiella pneumonia shows the tolerance limit of 1000, and minimum was for, you see, Bacillus thuringiensis, it was 600 ppm. And then what we did, and metal tolerance, it's a common process in many organisms which possesses different mechanisms like extended delay stage, adaptation to heavy metals, and SAM can be applicable to this study. In our study, presence of metal tolerant species of bacteria considered metallophiles is attributed to increasing expression of metallothioneins. It can be when we see their range of tolerance is been increasing, it can be due to the expression of metallothioneins. Actually, these are some of the proteins under the stress which are being secreted under the stress, and production of various enzymes is not affected by the presence of metals, suggesting that in metal tolerant bacteria, these enzymes are well protected and keep the function of the isolate active. There are certain enzymes which during the stress are not being affected, and when the enzyme activity of the microbial isolate or any microorganism is not affected, it keeps him active doing his normal functions. You see, we cannot rely out the extracellular polymerase EPS and binding, which binds these metals to these. These are some of the mechanisms behind the tolerance of the microorganisms. You see, then we have the sequestration potential, and we have all again taken the sequestration potential, how we sequestrated isolates were inoculated in broth having the concentration of 100 ppm of respective metal. All these metals were put in the broth, keeping the concentration of the metal 100 ppm. Positive controls were also run in that contains broth and inoculums of the isolates, only positive control. And we have then the 5 ml of the sample was taken different times, centrifuge with that, supernatant was immediately filtered through the filter paper 0.22 micrometer, micro filter, and then the supernatant was acid digested. We use the tri-acid digestion or di-acid digestion. Then we analyzed again the sample with the AAS, and we got the sequestration potential results. Among the metal tolerant bacteria, I would like to say, among the mercury tolerant bacteria, Staphylococcus ichneumonacracia, and you see that Pseudomonas putida, which show the highest potential for sequestration, that is 73% for mercury after 48 hours, where least was shown by Klebsiella pneumonia. And in case of cadmium, again we have Klebsiella pneumonia, which show the highest sequestration potential, and whereas Pseudomonas show the lowest. And among these Bacillus thuringiensis for the lead, it shows 65% of after 40, and Staphylococcus werneri show the least. And nickel, it was actually Klebsiella pneumonia, which shows the 58% of sequestration after 48, and Bacillus shows the least at 50% after 48 hours. This is the removal percentage after 0 hours, 6 hours, 12 hours, 18 hours, 24. After every 6 hours, we have taken the sample, analyzed it, and make it ready for the AS, and then we have analyzed it. Capital Pseudomonas showing 73% of the 48 for the mercury, and least was for Klebsiella pneumonia. And this is the graph showing the decrease in the content of the bacteria, this metal which we have given in the broth. And again, so for the cadmium, we have Klebsiella pneumonia with the highest sequestration potential, Pseudomonas putida with the least, and then we have graphical representation of this. Again, we have Bacillus thuringiensis and Staphylococcus aureus. And for nickel, we have Klebsiella pneumonia with the highest sequestration potential, and Bacillus saffronensis with the least. Many microorganisms synthesize extracellular polymers, and they bind the toxic metals, cations, and thus protect the delicate essential cellular compounds. This can be the one of the major this what you called as mechanism for showing the tolerance and for sequestrating of metals. There can be EPS, extracellular polymerase. There can be intrinsic properties. EPS composition is very complex. Sequestration of the several anions are known to react with different metals, which include chlorides, phosphates, hydroxyl ions, insoluble precipitates. Several microorganisms use lead to precipitation to lower the concentration of lead, and it can be done with number of mechanisms are being associates. And uptake of metals, we know that there are majorly two mechanisms. One is called as PACE2 mechanism, another is called as EC2 mechanism. Metal sequestration processes by extracellular sequestration and intracellular sequestration plays an important role in the removal of metals from contaminated environments. And results also revealed that certain bacteria in this study has a multi-tolerant metal resistance. Like we have Staphylococcus aureus showed tolerance to mercury and cadmium. Staphylococcus werneri that showed tolerance to mercury, lead, and nickel. Klebsiella pneumonia showed tolerance to mercury, cadmium, nickel. Pseudomonas putida showed tolerance to mercury, cadmium, nickel, and Bacillus saffronensis showed tolerance to mercury and lead. And so the conclusions from the study were that the concentration of heavy metals were found maximum in proposed study as compared to our control study. WHO participant limits for mercury and copper have been crossed, which is a matter of concern for us, especially the people of Jammu and Kashmir who are growing the saffron. Cement factories, use of pesticides are major source of heavy metals in the saffron soils. Bacteria belonging to diverse groups were present in saffron soils with Bacillus as dominant phylum. Our observation contribute to the understanding of metal tolerance among the different bacteria from saffron soils of Kashmir Valley, highlighted their role. With regards to Bacillus saffronensis in this study, it's the first report of lead tolerance. And for this bacterial species, a metabolism of resistance to heavy metal must be taken up for further studies. And this was the first report of lead tolerance. We have also we published that also. The metal resistant properties of this bacteria can be beneficial for detoxifying, eliminating heavy metals, thus providing an eco-friendly and sustainable option for to be used for bioremediation. That is, can be concluded that bioenvironmental interaction between bacteria and the metal is of great significance in a remediation of heavy metal contaminating soils and making it farmer friendly. So recommendations which I have put at the time of my on the basis that to study the impact of heavy metal on the growth parameters of saffron, it should be done. And additionally, synergetic action of plant microbe and their mechanism for metal mobilization, transfer, and detoxification should be studied. Use of metal tolerant bacteria for sequestration of saffron in metals in saffron affected soils. Despite several findings to the date, various steps of regulatory network like plant associated microbes in heavy metal citrus are still unknown. More investigation needs to be done for unveiling the crosstalk among the soil microbes and the metal interaction in different crops. Further experiments should be studied in natural conditions, and I told you this for the laboratory conditions to ascertain the exact metal removal capacity of the identified bacteria. Thus, we can the highlights of important consistent link between the research and development for assessment and treatment of emerging metal pollutants and tools, equipments, know-how that contributes towards the fulfillment of the challenge. Use of pesticides and herbicides in the saffron should be minimized. And you see, these are the two main important publications we have published. One of the paper in Frontiers of Microbiology last year. And we have also published one paper in Saudi Journal of Biological Sciences in 2010. It was 2020 and it was 2022. We were able to publish our this PhD data in my Frontiers of Microbiology. Other publications like we have in Journal of Bioremediation, some of the chapters, book chapters, and we have also published it. And these are some of the book chapters. These are conferences which I have attended personally, some important conferences. My studies will not be complemented, will not have been come to end because of the cooperation that has been provided by Department of Environmental Science, University of Kashmir, Director Cod, ex-Director Cod, and the faculty members. My sincere thanks to my both supervisors, extremely well talented and well knowledgeable persons two great scientists I have worked upon, Dr. Ruqayya Nazin and Professor B.A. Ganai, one of the two great scientists I have worked with. And my sincere thanks again goes to NCMR Pune for providing me the lab facilities for doing the molecular as well, the multitude of work. And thanks to DSG sir for providing some of the funds during my PhD. My special thanks goes to one of my mentor in the PhD, although he was not as my guide or co-guide. Still, without his guidance, my PhD would not have been completed. And that is Dr. Praveen Rahi, who have very much helped me during my PhDs, and all my lab mates. Sincere thanks to the microbiology faculty, Dr. Shaukat Ahmad, Dr. Javed Pare, Dr. Humayra Qadri, and to all my friends, unconditional support always by Said Mudassar Mushtaq, Ishfaq. And I will thank Microbial Society of India again for providing me an opportunity to present my work again on their platform. Very nice. I'm thankful to Ajay sir, Professor Deshmukh, and sir, Dr. Professor Sarvinjit Singh sir for providing the young talents the opportunity to present their work. Thank you. Thank you all.
Thank you very much, sir. Very nice and very interesting presentation. Thank you. Now we are open for question and answer sessions. So please ask your questions if anybody.
Sir, namaste, sir.
Morning, namaste.
Sir, I'm also doing research about the rhizobacteria present in metal contaminated soils or polymer soils and normal soils. So, how can I get much more details about types of tests or something, which tests we have to perform, and very advanced technique to observe or to identify new bacteria in these soils?
To absorb the new bacteria in the soil, we have to go for extensive culturing of the bacteria. That's one thing we have to go for. Second, if you want to go in depth of these bacteria, nowadays transcriptomics is a very nice field which we can work upon. Transcriptomics and metabolomics of the plant you can do, and I will suggest to from where you are working in your PhD topics.
Sir, I'm from Telangana, Hyderabad nearby.
Hyderabad have a very nice institutes. CCMB, I think CCMB is in Hyderabad.
Yes, sir. Yes.
There isn't everything is present in CCMB Hyderabad, and you can also go with touch with NCMR and NCCS latest sophisticated technologies. Like if you want to do the biochemical test nowadays, we have IV Tech and we have biochemical analyzers, and every facility is available in NCMR Pune. They are up to date. They have the sequencing facility available there. You can visit there. You can write from your guide to the lab of NCCS, NCMR Pune, and they can guide you very well. And for the identification of new bacteria, I will suggest you have to go for extensive culturing of the bacteria. That's the thing we can go for.
Thank you. Thank you very much.
Hello, Ali.
Yes, sir.
Did I meet you in Srinagar?
Yes, sir. You have presented me the Young Scientist Award in the Srinagar conference.
Right. And I met you, and I have your WhatsApp, many photographs.
I
Very
sometimes
nice.
text you also, sir.
Yes, I remember that.
Yes, sir. Yes, sir.
And we'll keep on in contact. We should be in contact.
Yes, sir. Obviously, there isn't
Yes.
any.
Right. Now, did you encounter some new species of bacteria?
Obviously, sir, I haven't encountered with new species from that. But what I recorded was the first report of Bacillus safinensis. I told in my presentation, this was the
Yes.
first report of lead tolerance.
Okay.
Yes, sir.
So any novel bacteria have you isolated? Any
Sir,
new?
I told you from novel bacteria, I haven't get any novel bacteria. Reason being from a novel point of view, the extensive work we did, and for this kind of work, I have to visit NCMR for
Right.
completion of these rest of the objectives, molecular identification, then multi-task and MIC. And I was thankful enough to be associated with Professor Dr. Praveen Rahi from NCCS Pune, NCMR Pune, who gives me an opportunity to work with him.
Right. Okay. Because you are working in a very, very nice site, that is Saffron field.
Yes, sir.
So Saffron will have some effect on the novel species.
Obviously, sir. I tried to convey. I tried to put my last year. I had applied for NPDF, SERP NPDF. I put the project for transcriptomics and metabolomics of the saffron whole transcriptome.
Right. Yeah.
But unfortunately, that was not been sanctioned and it remained that site.
You try to isolate some new species from the saffron field. It will be very interesting.
Yes, sir.
And now there are so many private companies that sequence the microbes.
Yes, sir.
Of course, there are CSI laboratories like CCMB and IMTech and many others, but you can also get it done in a private company.
Sir, sequencing was not an issue because Dr. Rahi told at that time when I left the lab, there isn't any issue if we send some of the isolates to the NCCS Pune or NCMR Pune.
Right,
they get
right.
sequencing. I have done the sequencing of next gen. Next gen these days was a new technique. Nowadays they use that and they give it. There isn't any issue. They provide every facility. The thing is only we have to write to them. They provide. Dr. I think the director has now been changed in NCMR. Previously it was Yogesh Soche, Professor Yogesh Soche.
I know. Achha, now all the battles can be analyzed by AAS.
Yes, sir. These can be analyzed using AAS.
Any metal.
Yes, sir. If we have that, what you called as, there's a probe for every metal. If we have that lamp, then we can analyze the metals on that.
What about mercury?
Yeah, mercury. We have to shift the that is VGA. Paper. We don't use the lamp. It is different from other metals. Yes.
It is a graphite furnace. Graphite furnace.
Yeah, we use the VGA system there.
Right.
Yes, sir.
Yeah, very interesting. Very good.
You have to assemble your AS in a different way.
Do you have a AS with you in your lab?
In University of Kashmir, I was having with that. But fortunately, after completing my PhD, I was associated with a project from DST SERB, where we get an AAS of in our lab, and I worked on that. And I'm an I will say I can use AAS very nice well.
Okay.
I use it to study from last 2 years. I use it AAS with my own, without any help. I got some training, basic trainings for AAS usage, and then I use it in the lab.
Very nice. You have done very good work and very well presented. Thank you so much.
Thank you, sir.
Thank you.
Thank you, sir. Do we have any more question in the audience?
I will request Ukab.
Yes, sir.
To give your email ID in chat box.
My email is okay, sir. I'm writing
in the chat box.
Okay, now Jay, you can conclude. No problem.
Okay, thank you very much, sir. Now, as Dr. Ali has put his email ID in the chat box, if anybody has any further questions, you can directly send an email to them. And thank you everybody for being here. Have a nice weekend. Thank you, Ukap, sir.
Thank you.
Very interesting lecture. Yes, we haven't received the mail ID, sir.
Sir, I have written, but I don't know why it is not going there.
You tell.
You tell, no worries. I will.
Please, you write down UQAB.
UQAB.
ALI.
Just a moment. ALI.
BABA.
BA. Right.
@gmail.com.
All right. I got it.
I also send it in the this.
Okay, perfect. We have it.
Yeah.
All right.
Yeah.
Thank you. Thank you very much. Thank you, Deshmukh sir, for providing us the opportunity. And thank you everybody for being here. Thank you, Ali sir.
Thank you for providing me an opportunity again.
Yes. Dr. Ukab, you will receive memento from our organization.
Okay, sir. Thank you. Thank you very much.
Within 15 days.
Okay, sir. Thank you. Thank you very much, sir.
Okay.
Okay. Have a nice weekend, everybody. See
Okay,
you on
thank you.
next Sunday.
Okay, bye.
Bye.